e-learning content of Sri Sharda College for Women. This content is about the economic importance of bacteria. Now, let us dig on to the economic importance of bacteria. My name is Gomathi. I teach students botany at Sri Sharda College for Women. Fine. Now, while coming to the economic importance, this involves both harmful aspects and useful aspects. Now, let us dig on to the harmful aspects first. To start with, deterioration of materials. Bacteria can decompose a variety of articles. The species of Mycobacterium, Pseudomonas, destroy cork, rubber, and cordage. They also destroy cellulose, the base of paper. Fouling of submerged naval carriers is also done by bacteria. Water pollution. Several bacterial forms cause water pollution. Say, for example, Vibrio cholerae, Salmonella typhi spoil the drinking water. Another hazard caused by bacteria is conversion of mercury compounds released by industries into methyl mercury. This compound reaches human body through food chain where it damages the nervous system. The famous Minimata disease is due to the methyl mercury poisoning. Denitrification. The breaking of soil nitrates into molecular nitrogen is called as denitrification. It is brought about by bacterial forms like Bacillus subtilis, Pseudomonas fluorescens, Thiobacillus denitrificans, etc. As a result, the soil fertility is reduced. Spoilage of food and food poisoning. Bacterial forms like Pseudomonas, Escherichia, Micrococcus, Salmonella, Staphylococcus, Clostridium, etc. They spoil foodstuffs and cause food poisoning through endotoxins and exotoxins. Marine foods such as seaweeds, oysters, crabs, and fishes are destroyed by Spirillum rectificitaris. Bacteria also causes diseases in plants, animals, and human beings. Say, for example, bacteria causes several diseases in plants such as galls, canker, leaf spot, blights, soft rots, etc. Some of the important plant diseases caused by bacteria are the crown gall in many plants caused by Agrobacterium tumefaciens, tondu of wheat caused by Cornybacterium triticum, soft rot of mango caused by Erwinia carnivora, red stripe of sugarcane caused by Pseudomonas rubelianensis, citrus canker caused by Xanthomonas citri, etc. Further, Bacillus+anthracis causes anthrax in horse, mule, and elephants. Mycobacterium+tuberculosis causes tuberculosis in cattle, dogs, and parrots. Besides, different species of Salmonella like Salmonella dublin, Salmonella pullorum causes abortion in domestic animals. Further, some common diseases such as anthrax by Bacillus+anthracis, bacterial food poisoning by Bacillus cereus, diarrhea by Bacillus coli, botulinum by Clostridium botulinum, diphtheria by Cornybacterium diphtheriae, influenza by Haemophilus influenzae, tuberculosis by Mycobacterium tuberculosis, leprosy by Mycobacterium leprae, gonorrhea caused by Neisseria gonorrhoeae, typhoid caused by Salmonella typhi, dysentery caused by Shigella dysenteriae, cholera by Vibrio cholerae, and plague like Yersinia pestis are some of the common diseases caused by bacteria in human beings. Human diseases like dysentery, cholera, paratyphoid, typhoid, gastroenteritis, etc. are caused by taking contaminated food or water or some fecal contamination On the other hand, diphtheria, influenza, pneumonia, tuberculosis, and whooping cough are caused by droplet infection, that is, by the dispersion of droplets into the air from an infected respiratory tract. Plague and tularemia are diseases of rodents. While plague is transmitted to human beings by flea bites, tularemia is transmitted by tick bite. Gonorrhea and syphilis are transmitted by sexual contact, as also from infected mother to fetus. Biological warfare. Pathogenic bacteria, such as those causing anthrax, botulism, cholera, tuberculosis, etc., are also used as very strong weapons to be used against the enemy. Their effects are long-lasting and much far-reaching. There is anthrax fever around the globe, particularly in USA and UK in their 2000 war against Afghanistan. Postal transmission of anthrax has been recorded as new phenomena during those days, and deaths due to this disease have also been recorded in the United States of America. Well, now moving on to the useful aspects. About 2,100 antibiotics have been reported so far from actinomycetes alone. Of these, maximum antibiotics have been reported from streptomycins alone. Now, some of the common antibiotics are gramicidin and tyrocidin caused by Bacillus brevis, bacitracin and licheniformin by Bacillus licheniformis, streptomycin by Streptomycetes griseus, aromycin by Streptomycetes aurifacius, erythromycin by Streptomycetes erythraeus, neomycin by Streptomycetes fradiae, terramycin by Streptomycetes ramosus, chloromycetin by Streptomycetes venezuelae, etc. While kanamycin and polymyxin B are effective against gram-negative bacteria, the rest are effective against gram-positive. Audomycin and chloromycetin are effective against both. The streptomycin is generally used against TB, meningitis, and pneumonia, while the aromycin against osteomyelitis, whooping cough, etc. Moving on to soil fertility. The bacteria are important agents in maintaining soil fertility by nitrogen fixation, ammonification, and nitrification. The fixation of nitrogen is brought about asymbiotically by Clostridium, Acetobacter, Chromatium, Rhodomicrobium, Rhodospirillum, Rhodopseudomonas, Spirillum, etc. While Clostridium is an anaerobe, Acetobacter is aerobe. Symbiotic nitrogen fixation is brought about by Rhizobium in the root nodules of legumes, actinomycetes in the root nodules of Myrcia, Alnus, and Casuarina, as also by Klebsiella in the leaf nodules of Psychotria, Pavetta, and Dioscorea. The ammonification, breaking of proteins into ammonia, is facilitated by Bacillus ramosus, Bacillus vulgaris, and Bacillus mycoides, etc. The nitrification, conversion of ammonia to nitrate, is brought also by Nitrosomonas and Nitrobacter. Moving on to the industrial uses, acetic acid is produced by the bacterium Acetobacter aceti from ethanol. Clostridium acetobutylicum converts acetic acid into acetone and butyric acid to butanol. Streptococcus lacti converts sugars to lactic acid. Acetobacter is used for producing substrate, which yields butylene glycol used in production of synthetic rubber. Propionibacterium is used in the manufacture of propionic acid. Brevibacterium, Cornybacterium, Micrococcus, etc. are employed for glutamic acid fermentation. Cornybacterium glutamicium produces the amino acid lysine. Escherichia coli produces the amino acid threonine. Clostridium acetobutylicum during sugar fermentation also produces vitamin B. Flavoring of tobacco and tea leaves is done by Bacillus megatherium. Coming to retting. Commercial fiber is separated in the fiber-yielding plants by the decomposition of non-cellulosic cementic materials through a process called retting. It involves microbial decomposition of unwanted material. Retting of flax, jute, and hemp is done by bacterial forms like Clostribacterium butyricum and Clostridium falcineum. The milk products such as butter, cheese, curd, etc. are obtained by the action of bacteria. The milk contains bacterial forms like Streptococcus lacti, Escherichia coli, Lactobacillus lactis, Clostridium species, Pseudomonas species, etc. Yogurt is prepared by Lactobacillus San Francisco, Lactobacillus bulgaricus, and Streptococcus thermophilus. Well, moving on to the environmental protection, bacteria are capable of decomposing some hazardous pesticides such as 2,4-D by Acramobacter, Flavobacterium, 2,2-dichloropropiononic acid, Dilapone by Agrobacterium, and DDT partially by Acetobacter aerogenes, etc. Biological control of some insects is done by bacteria. Say, for example, Bacillus papillae kills Japanese beetle. Bacillus spiricus kills malarial mosquitoes. The disposal of sewage is also done by microbes, including bacteria. The material cycles in an ecosystem are completed by microbial bacterial activities. Fermentation of urine to ammonia and carbonic acid compost and silage production is also materialized by bacteria. Methanobacillus occurs not only in marshes but also in dung. It produces methane under anaerobic condition by fermentation of organic matter, which is the source of power. In the process of tanning hides, the tannins are broken down by bacterial fermentation. This protects hides from spoilage by putrefaction. Thiobacillus peroxidans, an anaerobic bacterium, oxidizes sulfur, sulfides, and iron. Iron pyrides occur in nature in association with uranium and copper. This bacterium is capable of increasing the dissolution rates of pyrites by million times, which is otherwise insoluble. The bacterium cannot act upon oxides, but the mineral can be obtained bacterially from sulfates. The isolation and transfer of foreign DNA segment gene into a new host is genetic engineering. The most useful technique for achieving the above goal is called recombinant DNA technology. The plasmid acts as a carrier of gene. The tiny plasmid of Agrobacterium tumefaciens has very much exploited by researchers.